Hello everyone, in this video I am going to show you how to design PCR primer step by step. So first let's know about the PCR process. So in the PCR process is basically, let me use the laser. So a PCR process basically conducted in the appendrop tube, which is basically shown here. And in the PCR process there is basically 3 to 4 main phases, and the first phase is denaturation. And in the denaturation phase, basically the double stranded DNA unwinds and becomes single stranded DNA, and the temperature in the denaturation phase is around 94 to 96 degrees Celsius. And then the next phase is annealing, and the annealing temperature is basically 55 to 60 degrees Celsius. And in this stage, the the single stranded the primer, basically here the green color strand is basically primer. So in the annealing stage, the primer is basically binds with the unwind DNA or binds with the single stranded DNA. And then the extension phase, and in this phase the DNA polymerase enzyme basically binds with the 3' end of the primer, and it elongates the template DNA or elongates the newly formed DNA. Okay. And this is basically the main process of PCR. So a PCR requires 2 primers, a forward primer and a reverse primer. And basically primers are short DNA sequence that bind to opposite strand of the target DNA, and it provides a starting point for DNA polymerase to synthesize new DNA strand, which we can see here that the DNA polymerase enzyme basically binds with the 3' end of the primer. Let's say this is the 3' end and this is also the 3' end. So the DNA polymerase binds with this 3' end, and then it elongates the newly formed DNA. Okay. Now let's talk about some key criteria for designing good primer. So first of all, the primer length, and the primer length should usually be between 18 to 25 nucleotide or 18 to 30 nucleotides. And this range is basically provides a good balance between specificity and efficient binding. Second, let's talk about the GC content. The GC content should usually be between 40 to 60%, and a balanced GC content ensure proper stability of the primer template interaction. So if the GC content is far more than 60%, then a hairpin loop can form or a primer dimer can form. So we should maintain this range around GC content percentage should be maintained in between 40 to 60%. And third, consider the melting temperature, and the melting temperature for both primer basically forward primer and reverse primer should generally be between 55 to 65 degrees Celsius or 50 to 65 degrees Celsius. And the difference between the the difference between the forward and reverse primer should not exceed 2 degrees Celsius or 3 degrees Celsius. We have to maintain this, that the difference between the forward primer melting temperature and the reverse primer melting temperature should not uh, be exceed should not exceed 2 degrees Celsius or 3 degrees Celsius. And another important feature is the GC clamp, and GC clamp is basically the this part, the end part or 3' part of the primer. Basically this is basically called GC clamp. So uh, having one or two guanine or cytosine nucleotide at the 3' end of the primer can improve the binding efficiency during the PCR. So the GC clamp should be 1, 2 or 3 pairs at the end of the 3'. And next we have to also determine the desired amplicon size. For conventional PCR products, between 100 to 1000 base pairs are commonly used, and while for qPCR, shorter amplicons ranging from around 70 to 200 base pairs are also preferred. Now, to simplify the process, we can use directly Primer-3, or we can use the NCBI, because NCBI is basically integrated with the Primer-3, and Primer-3 is basically best for primer design. So let's go into the NCBI. Let's go into it, and let's select the gene. and let's search for a gene. And from here you can also select the organism. Let's select Homo sapiens and just wait for few seconds. And let's say you want to work with this sequence, so just click on it. Or you can also open in a new tab. So from here, scroll down and select the RefSec RNA. So select this or open this in a new tab. Okay, now just select the first sequence here. So from here you can directly click on pick primers, or you can also click on this highlight sequence feature. So basically from this highlight sequence feature you can exactly see the exon sequence or the coding sequence of your mRNA. So just wait for a few seconds here. Okay, so from here you can see that this is the whole mRNA sequence, and from this whole mRNA sequence the selected part is basically the coding sequence, CDS. Or you can also select the exon. So this is exon, this is also exon, this is also another exon. So those are the exon sequence of this whole mRNA, and this is basically the whole gene. So yeah, this is the poly A site. So from here you can select, or you can choose which sequence you want to amplify. Okay, so just go into the previous tab. Just click on the pick primer. So now you can see that the accession number is already appeared of that sequence, and you can also select the forward primer or the reverse primer range. So basically for this reason, for the selection process, you obviously have to study about the sequence, that which part you want to amplify. Okay, so then just okay, let the tab load perfectly. Okay, okay, now the tab is basically perfectly loaded. So from here just keep everything by default, or you can change this a little bit. Let's say primer melting temperature 55, and the maximum is let's say 65, and you can just keep this as it is, and select this as NT, and select this as primer must span an exon-exon junction. For this, the primer is basically appear in less contaminated with the genomic sequence, and this is the basically Homo sapiens accession number. And then you can also select the advanced parameters, and from advanced parameters you can select the PCR product melting temperature, primer size, let's say 18 to 20, let's say this 30, make this 30, and GC content here, let's make this 40, and this make this 60, and GC clamp, let's make this 2. uh, so the primer is basically strongly binds with the 3' with the DNA sequence, and keep everything by default, or okay, just keep everything by default. Click on this show result in a new window and click on get primers. Okay, after appearing this step, just scrolling down and just click on submit. So it basically blast with the sequence we selected. So it basically blast with this sequence. So after blast, just click on submit and the PCR, then the PCR result will appear. Okay, so the PCR result has come. So let's see. So from here you can see the PCR one, sorry, so from here you can see the primer 1, primer 2, primer, or all the predicted primers. And from here you can download the primer pairs in CSV format or in text format or in tabular format. So let's suppose that this is primer pair 1, and if we see that the length is okay, 21 and 20, and start with this and stop with this sequence, and the melting temperature is 60, and this is a good melting temperature because we know that the difference between the melting temperature is basically lesser difference is basically good, and higher difference is basically, or big difference is basically bad for a primer. And GC content is also decent. GC content is in the range of between 40 to 60, and self complementary, and this is a issue that this is not that much issue because the self complementarity threshold value is globally 8. So here the reverse primer self complementarity is 5, and this is 3, and self 3 prime complementarity is also good, 1 2, didn't exceed the 3. So overall we can say that the primer pair 1 is good. And here, from here you can also see the other primer pairs. Just you can just click on it and you can see the details of that primer. Okay, so yes, yeah, that's all for today's video. If you find this tutorial helpful, don't forget to like the video, subscribe the channel, and turn on the notification bell. Thank you for watching.